Welcome back, everybody. Now we are on the fourth video of the chapter on coordination, and this is A2 biology. And the syllabus code is 9700. And I hope you're all enjoying this chapter, just like I'm enjoying making these videos. This video is a continuation of the previous three videos. The topics covered here are number one, steps of muscle contraction, which is the famous sliding filament model, and we're going to be discussing that. The next topic is hormones and the menstrual cycle. Now, this is all part of coordination, as you remember, the nervous and the endocrine coordination. And then there's a little short bit about birth control. First of all, let's look at the structure of myosin and actin. Now, myosin is the thick filament, and it has many molecules of myosin, but please remember myosin is a fibrous protein, but it has globular heads, and these globular heads are these. So the globular heads, and look at the globular heads, they point away from the M line. Now this is the M line. So they point away from the M line. Now this is very important that you understand the structure of myosin, which is the thick filaments. So it's a fibrous protein, but it has these globular heads. Please do not mix this with globular proteins. So globular heads, thick filaments, and they point away from the M line. The next thing that we need to study is the structure of actin. Now, important thing to remember is that actin is the thin filaments, but actin is a globular protein, and actin has many actin molecules are linked together to form a chain, and it's like a string of pearls. And two of these are twisted around each other. Like I've just drawn this in blue, so this one is twisted around this other one, which is this other second one. So like two strings of pearls are twisted around each other. So this is one and this is two. And these are these actin molecules which are linked together to form a chain. So I'm keeping it to the two blue lines which I've drawn here. So this is the one line, and then the second blue line, which you need to understand. So this is one blue and another blue twisted around it. So these are the two actin molecules. And then we have another fibrous protein which is called tropomyosin. And the tropomyosin is somewhere twisted in between these two actin molecules. And then there is a fourth which is called troponin. Now troponin is not continuous, it is found at regular intervals. So this is one, this is the second, this is the third, this is the fourth, and this is the fifth. So troponin molecules are not continuous and they're found at regular intervals. So again a recap, the two actin molecules twisted around each other, then the tropomyosin in between twisted, and then the troponin at regular intervals. So we are talking of four. So we are talking of two of the actin molecules. This is the one, two of these. Then one is the tropomyosin, and the fourth is the troponin. Please, everybody must remember the two actin molecules twisted around each other, then the tropomyosin, which is also twisted in between the groove of the actin molecules, and then we have at regular intervals the troponin, which I have colored in green. Now a quick recap of how muscles contract. First of all, the calcium ions are released from the sarcoplasmic reticulum. Then these calcium ions bind to troponin. Now remember, troponin was those which are found at regular intervals. Then the troponin and the tropomyosin move. When they move, they expose parts of the actin molecule. Now the myosin, which was the thick, the dark bands, the myosin heads then bind to the actin, and they form a sort of a cross bridge. They form a sort of a cross bridge. This is a cross bridge. Myosin heads then tilt and they pull the actin. Then the heads hydrolyze ATP. That's why a lot of ATP is needed for muscle contraction. Then the heads let go of the actin. Then they bind again to the exposed site. So now the thin filaments, which were the actin, have moved. Then the myosin heads bind to the actin again, further along the thin filament. Now this would be more closer to the Z line. Then they tilt again, pulling the actin, hydrolyze more ATP, and of course this goes on and on, so long as the muscle has a supply of ATP. So the muscle contracts, and this is called the sliding filament model. This is a very nice diagram showing this sliding filament model, and as you can see the two actin molecules twisted around each other, then the tropomyosin which is twisting around it, this the one in the red, and this regular thingy is the troponin. You can see the troponin is very clearly, you can see the yellow structure which is the troponin. So this is the troponin, this is the troponin here, this is the troponin here, this is the troponin here. So as you can see, the first one, the myosin heads attaches to the actin myofilament, forming a cross bridge. Then number 2, inorganic phosphate generated in the previous contraction cycle is released, initiating the power stroke. The myosin head pivots and bends as it pulls on the actin filament, sliding it towards the M line. Then ADP is released. Number 3, as new ATP attaches to the myosin head, the link between myosin and actin weakens and the cross bridge detaches. Number 4, as ATP is split into ADP and inorganic phosphate, the myosin head is energized, cocked into the high energy conformation. And then of course, one again, myosin head attaches to the actin myofilament, forming a cross bridge. So this is the sliding filament model. Maybe you can pause the video here and have go through it again, and please, you need to learn this. This is another diagram which again explain this to you in a slightly different manner. So number 1 is the exposure of the active sites before cross bridges cycle. Calcium binds to the troponin. You can see the calcium, you can see the troponin. Calcium binds to the troponin and the tropomyosin moves. So calcium binds to the troponin, tropomyosin moves, exposing active sites. Number 2, cross bridge formation. The myosin head binds to the exposed active sites on the actin myofilaments to form cross bridge, and phosphates are released. Number 3, power stroke. Energy caused in the myosin head is used to move the myosin heads, causing the actin myofilament to slide past the myosin myofilaments, and ADP molecules are released from the myosin heads. Number 4, cross bridge releases an ATP molecule binds to each of the myosin heads, causing them to detach from the actin. Then number 5, hydrolysis of ATP, the myosin ATPase portion of the myosin head. Split ATP into ADP and phosphate, which remain attached to the myosin heads. Then number 6 is the recovery stroke. The heads of the myosin molecules return to the resting position and energy is stored in the heads of the myosin molecules. If calcium is still attached to troponin, cross bridge formation and movements are repeated. This cycle occurs many times during a muscle contraction. Not all cross bridges form and release simultaneously. So you need to go through this once more and see how the muscle contraction is occurring. Now we're going to be discussing the neuromuscular junction, and you can see here the labeling here. This is the sarcolemma, which is the cell membrane. This is the T-tubule, which are these tubes which are in the holes in the membrane, which are sort of invagination of the cell membrane. So the T-tubules are more the invagination of the cell membrane. You see the cell membrane is this blue structure, and this sort of forms a tube inside it. And this is called the T-tubule. So if you look at the first thing is net entry of sodium ions initiates an action potential which is propagated along the sarcolemma and down the T-tubules. Now the action potential in the T-tubule activates voltage sensitive receptors which in turn trigger calcium release from the terminal cisternae of the sarcoplasmic reticulum into the cytosol. So now these calcium ions were all stored here. So the calcium ions are all coming out of this sarcoplasmic reticulum. SR stands for sarcoplasmic reticulum. Now the calcium ions bind to troponin. Troponin changes shape, removing the blocking action of tropomyosin. Actin active sites exposed. Number 4, contraction. Myosin heads alternately attach to actin and detach, pulling the actin filaments towards the center of the sarcomere. And the release of energy by ATP hydrolysis powers the cycling process. Then number 5, removal of calcium by active transmode back into the sarcoplasmic reticulum after the action potential ends. Number 6, tropomyosin blockage restored. Blocking myosin binding sites on actin, contraction ends and muscle fiber relaxes. So this is how we've got to understand how muscles contract and relax. This is once again the events at the neuromuscular junction, and this is taken from the Cambridge reference book. Events at the neuromuscular junction: action potential arrives, action potential causes the diffusion of calcium ions. Calcium ion causes vesicles containing acetylcholine to fuse with the presynaptic membrane. Number 4, acetylcholine is released, diffuse across the synaptic cleft. Number 5, acetylcholine binds with receptors in the sarcolemma. Sarcolemma, please remember, is the cell membrane of the muscle, causing them to open channel proteins for sodium ions. Sodium ions diffuse in through the open channels in the sarcolemma. Please remember the muscle cell membrane is called sarcolemma. This depolarizes the membrane and initiates an action potential which spreads along the membrane. Number 7, the depolarization of the sarcolemma spreads down the T-tubule. Number 8, channel proteins for calciums open and calcium ions diffuse out of the sarcoplasmic reticulum. Number 9, calcium ions bind to troponin. Tropomyosin moves to expose myosin binding sites on the actin filaments. Myosin heads form cross bridges with thin filaments and the sarcomere shortens. So we have to be very clear how the entire process is going to take place, how the motor neuron reaching the muscle. There's going to be the same synaptic transmission over here, and this is the same as what happens in a synapse, and then of course what happens inside the muscle. Now we talk about hormonal communication. Now, in hormonal communication, just like we talked about the nervous communication, we do not have any nerves, but we have a blood supply which is going to facilitate this hormonal communication. So, the hormones that we have discussed previously have been adrenaline, glucagon, adrenaline produced by the adrenal glands, glucagon and insulin produced by the islet of Langerhans in the pancreas, ADH produced by the pituitary gland. Now, all these hormones are produced by glands, and the glands we say are called endocrine glands. Endocrine means ductless glands, just like we have certain glands with ducts, like the salivary gland has a duct. So the saliva is produced in the salivary gland and is transported through the duct, which is a tube, a channel, a pathway through which the saliva is then released into your mouth, and that keeps our mouth moist and we can talk. So that's why especially people who are debaters will always keep a glass of water because their mouth dries up. So because there's less saliva being produced because of the effect of adrenaline. So endocrine glands are ductless glands and they produce and release one or more substances. Like the islets of Langerhans produces the insulin, and there are two types of cells there, alpha cells and beta cells. So they produce insulin and glucagon. Now this process of producing and releasing substances is called secretion, and this secretion is going to be thrown into the bloodstream. So the blood is going to be the communicating fluid. Just like if you throw something into the river and then something down the river, somebody picks that stuff up. So there's a lot of wood is transported like this in certain northern areas of Pakistan, in which logs of wood are thrown into the river and then they're collected downstream. So secretion. So this is what we have to understand about the hormonal communication. Now the next thing which we have to understand is that hormones are cell signaling molecules. Now, usually there are two types of hormones. One which are the peptides, and these are proteins, and these are globular proteins. And you remember globular proteins are the hydrophilic R groups are on the outside and hydrophobic R groups are on the inside. So they are water soluble and they will bind to, so there's a cell here, then there'll be a receptor on its surface. And this is where the hormone will bind to it. So, and receptors, remember, are glycoproteins or glycolipids. So, water-soluble peptides, because they cannot cross the phospholipid bilayer, will bind to receptors, on the target cells. And these then activate a second messenger, and then they transfer the signal throughout the cytoplasm and results in some sort of a transcription and translation, which results in the formation of some protein molecules, which may be needed for the next steps of the reaction. The second type of hormone is lipid soluble, which we're going to discuss now in this coming topic that we're going to discuss. Now, the lipid soluble hormones will not have any receptors. So there will not be any receptors on the cell membrane, as I've just mentioned. So no receptors on the cell membrane, and these lipid soluble can pass through the phospholipid bilayer. So they will enter the cytoplasm and then they will bind to receptor molecules either inside the cytoplasm or even maybe on the nucleus. So the receptor molecules for these, please remember, very, very interesting questions have come in your papers on this, in which they will say, okay, why does it not have a receptor? Because it is lipid soluble. It's a hormone which is lipid soluble, so it can pass the phospholipid bilayer. So it will bind to receptor molecules inside the cell because the cell membrane is not preventing its entry. While the water-soluble hormones will prevent the entry, so they have a receptor on its surface. Now, this is the diagram that I've made for you. So what you need to understand is that lipid-soluble hormones will pass through the phospholipid bilayer and enter the cytoplasm. And inside the cytoplasm, there would probably be receptors for it, and maybe even on the nucleus, there would be receptors for it. And then it will activate transcription. So, cell signaling molecules, hormones. Next, we come on to the topic of hormonal control of the human menstrual cycle. And this is, of course, a topic which is very misunderstood. I teach it at all levels as well, and I find students really struggling with it. and not being able to understand it. And just before the exams, I have lots of queries about this. So I'm going to try my best to explain it to you as best as I can. We're going to be talking of the human menstrual cycle. So we're going to talk about the reproductive organs. Now, the first one is the uterus. The uterus is a hollow organ, and its wall is made up of a muscle. And inside is a layer which we can call the lining of the uterus, or we can also call it, the technical name is endometrium. Please remember, endo is always like we talk of endothelium, which is the lining of blood vessels. So endometrium is the lining of the uterus, and this endometrium is shed, and that's when when a woman has her periods or she menstruates, and this lining is shed. Please do not say the uterus wall is shed; that cannot be shed. So the uterus lining is shed, and this endometrium can also be called the lining of the uterus. So what you need to understand is that there is the uterus, which is a hollow organ, and the wall contains a muscle in it. Then it has two tubes which are attached to it, which are called the oviducts. Now the other name for the oviducts is fallopian tube. We can also call that. And then we have the two important organs, which are the ovaries. And there's two ovaries, one on either side. And the ovaries are the where the gamete is going to be made. The gamete is the, so we understand meiosis is going to take place here, and the gamete will contain haploid number of chromosomes. So the gamete by meiosis, and that is the ovum, is going to be produced here. We've talked about this in an earlier chapter as well. So ovum, gamete, meiosis, haploid, right? Now, what we have to understand in the human menstrual cycle is we're going to be considering, number one, the ovaries, what happens in the ovaries, and number two, what happens inside the uterus during this 1 month cycle. The 1 month cycle is the menstrual cycle, which is going to be discussed in more detail, and we're going to discuss the effect of hormones during this menstrual cycle. First, let's try to get an overview of the menstrual cycle, and I find this sort of a pie chart is very interesting in how you can see it. It's usually a 28-day cycle. So if a woman has started her periods on the 1st of January, so she's going to start her next cycle by the 29th of January. So it's a 28-day cycle, and we divide it into a number of different portions of it. Now, the first 5 days is when the uterus lining breaks down and menstruation occurs. Then day 6 to day 10, uterus lining thickens again. So the lining inside the uterus thickens again. Then we have this part, which is the ovulation occurs usually on day 14 of the menstrual cycle. And as we talked about 28, so day 14 is the ovulation date. But please remember, this is not so regular. Some women have a 28-day cycle, some even have a 30, 32-day cycle. So it varies from women to women. And then after ovulation has taken place, now two things can happen. Ovulation means the release of the ovum. So the release of the ovum takes place. Now, two things are possible. Number one, fertilization takes place. So the woman is pregnant now. So now after this, there'll be no more menstruation. There'll be no menstrual cycles. For 9 months till the baby develops or the fetus develops inside the uterus, there's going to be no menstrual cycles. But normally, I mean, a woman may have 3 children or 4 children, but all her menstrual cycles are usually without any pregnancy. So mainly we have to understand what happens if no fertilization takes place. Then, of course, the normal event is the uterus lining continues to thicken. This is till day 28. So uterus lining continues to thicken because it is anticipating that maybe a pregnancy takes place. So then it is going to come and implant here. What is going to implant is going to be the zygote, because if fertilization takes place, the sperm and the ovum fuse and a zygote is formed. So this is the overview of the menstrual cycle. Number one, uterus lining breaks down, first 5 days, then uterus lining thickens, then ovulation occurs, then uterus lining continues to thicken, because if pregnancy takes place, then it has to implant, it has to embed. But if by day 26, 27, 28 nothing has happened, then the uterus can figure that out and it prepares to then start the next cycle will start and the uterus lining breaks down and menstruation occurs. Now, the two organs that we are going to consider in the menstrual cycle are the ovary and the uterus. Now, what is happening inside the ovary? Now, that's very important that we understand it's only got divided into two phases. The story in the ovary. The ovary, the first story is number one, a potential female gamete starts to develop. A primary follicle is produced by and developed tissue surrounding the developing gamete. So we have the gamete in the center and we have the follicle cells around it. And this, of course, becomes larger and larger and larger. And then some of the cells liquefy and it forms a fluid filled space and still attached to the main. And this whole story, this whole thingy is called a Graafian follicle. So that is why in some books it is also called the follicular phase. So this is the Graafian follicle. So the Graafian follicle develops from day 1 to day 14. And then the primary follicle becomes a secondary follicle. The secondary follicle develops into an ovarian or a Graafian follicle. Now on day 14, what has to happen is ovulation. Ovulation means that this is going to burst here and this part of it is going to be released and this will come, we see it outside with the gamete in the center. Now what is left behind? The remains of the Graafian follicle which are left behind now is not going to be called a Graafian follicle. Now this is now going to be given a separate name. This name now is not Graafian follicle. The same Graafian follicle without the ovum and the remaining follicle cell which has now come out of it. Now this has come out of it. Now this remains of the Graafian follicle, just by giving it another color, this is going to be called the corpus luteum. It was the same structure, but now it is going to be called corpus luteum, and this produces progesterone. Previously, when it was the Graafian follicle, it was producing estrogen. So the same Graafian follicle, which is now called corpus luteum, is going to be producing progesterone, and this happens on day 14. Day 14, ovulation occurs. Now, as it says here, the remaining tissue forms a corpus luteum, and this remains till the end, and then of course it degenerates and dies. Next, we talk about what is happening inside the uterus. Now, in the uterus, most of the endometrium lining is lost during menstruation, the first 5 days, day 1 to day 5, and then day 6 to day 13. The endometrium develops, ovarian follicle produced, and then we have, this is number 3, there's a mistake in the book. So the endometrium is most developed, contains many blood vessels, and here is when on day 14 ovulation takes place. And then of course the endometrium is maintained until corpus luteum degenerates, and then ovary corpus luteum develops and then degenerates. So this would be day 15 to day 27 or 28 even. So this is what is happening inside the uterus, and these are the changes which are leading up to and following evolution, following ovulation. What we need to understand is that what is going to happen after this is we're going to study the effect of different hormones. Now, this will continue, the effect of hormones will continue in the next video. So please look up the next video, which will have the hormones, how they control the menstrual cycle. Thank you. Thank you for watching.